Next, we look forward to the presence of Dr. Avinash Jain. A warm welcome, sir.
So thank you for the reason why I was looking forward to this program is it was actually a revision for us also, except for 2 to 3 topics that were given to us. And now we are going to move to some specific topics. We will be focusing more on each of the components of these innate and adaptive immunity. And these are my panelists. So if you have any questions at the end, you know whom to contact, right? A crosstalk with cytokines, and look at his menu card, thymus, interferons, look at his plate. It's all full of immunology. So you know whom to catch hold of if you have doubts. And can anyone tell why in 2020 interferons in India were very angry? Not exactly. This is what was happening, right? They were trying to drive away the virus with this simple mechanism. So, bichare interferons were feeling a little left out, and but we will see about what they do and what are the mechanisms behind it, the lack or the excess, what really happens, right? And this at the end of the day, probably we'll be able to realize why some patients were getting severe COVID or life-threatening COVID as against some who were asymptomatic or were only getting mild infection. So interferons are like wives. They would love to interfere, but it is context dependent. Sometimes the moods are good. They will help you kill the virus. Sometimes they can be really bad, and it might be detrimental to your own health. So that's how interferons are. They interfere. And where do we place them? So this is how the cytokine family looks like, right? This is just an overview because you will be hearing lots about cytokines, so you should be able to place these cytokines somewhere. So we have interleukin-1 family. We have class 1 hematopoietin family, which includes a series of cytokines. We will be focusing today on class 2 cytokine family, which includes interferon alpha, beta, gamma, and so on and so forth. We have the TNF family, IL-17, the chemokines, and this is not working, but can anyone tell which was the first cytokine to be discovered? Anyone? You want to take help of the slides here? Since I am taking the first lecture on a cytokine, so it was actually interferon which got discovered for the first time. And this is how they were, the name came. They were trying to grow this live influenza virus on a chick egg chorioallantoic membrane. And there was something which was interfering with the growth. And that's what they named it as interferon, interfered with growth. And that's how the naming ceremony happened for this. So this will be my layout today. Type of interferon, how they function, how they are going to kill the viruses, how that function helps us to learn their importance in health, and then role in diseases. That is, even extra of everything is bad. So this is how the family looks like. We have the type 1 interferon, which includes interferon alpha. There are 13 subtypes. Interferon beta, there is only one subtype, but I think as time is passing, this family is going to get only bigger. The most important role of interferon alpha and beta is killing the virus, helping in attacking the virus-infected cells, and they are secreted mainly by the macrophages and dendritic cells. And they are going to act on multiple cells, a number of cells they will act upon, those who are expressing the interferon alpha receptors, which we will see. Type 2 is interferon gamma. This family includes mainly interferon gamma. They are secreted mainly by the T and NK cells, and they act on macrophages. We will see on the B cells, on the CD8 T cells. They activate the macrophages, they drive the T cell to Th1 subtype. They also help in differentiation of cytotoxic T cells. And finally, we have, the lambda family, interferon lambda 1, which is called as 29, interferon lambda 2, which is called as 28A, and interferon lambda 3, which is called as 28B. They are mainly present, they mainly act on the epithelial cells, and they upregulate expression of genes, but more and more research are now showing role of these different interferons, they're crossing the boundaries. It's no more, limited just to one kind of work that we have learned so far. So this is as simple as it gets. A cell which is infected by a virus, this is the nuclear material, they get activated, they will start producing interferons. These interferons in an autocrine and paracrine fashion will help us to take care of this viral infection. Right? Now let us complicate the steps one by one. So this is a busy figure. First, let's start focusing on this side. I think this might not be visible, but this is a Baltimore classification for virus. Group 1, Group 2, Group 3, so on. They have divided the viruses into positive strand, single ssRNA, dsRNA, right? Negative strand ssRNA, dsDNAs. Why they have divided like that? As we were discussing, these pattern recognition receptors can recognize only particular set of viruses. Some of them will recognize dsDNAs, some of them are going to recognize ssRNAs, and so on and so forth. So this is how the story starts. A virus particle which has infected a human body, the cells are going to recognize these patterns which are reflected on the either surfaces of the virus, like envelope proteins, virus proteins will be recognized by, so this is the membrane. These are the pattern recognition receptors located on the membrane. They are trying to recognize the foreign particle. Then there are some of them which are pattern recognition receptors which are located inside the cell. Either they can be in the endosome, for example TLR9, the heterodimer of TLR7 and 8, and homodimer of TLR3. And if you look closely, they all have their own specific patterns that they have to recognize. Sabko homework mila hua hai, and that's what they are good at doing. TLR9 is recognizing CpG DNAs and viral DNAs. TLR7 and 8 is trying to recognize the single-stranded RNAs. TLR3 is recognizing the double-stranded RNAs. Then there are some cytosolic pattern recognition receptors called as MDA5 or the REG1, which is retinoic inducible gene 1. They are recognizing the DNA materials which are lying in the cytosol. Once they have recognized this foreign material, they are going to activate the cells here inside the nuclear material. The interferon responsive genes, that is interferon response elements, will get activated. Type 1, type 2, type 3 interferons will be formed. Like I said, excess will result in autoimmunity and a lack of it will result in higher susceptibility to a particular infection. So now let us focus on these steps gradually, what exactly is happening. So this is a more detailed version of those pattern recognition receptors I was talking about and their downstream pathways. The DNAs, RNAs are being recognized by these molecules called as cGAS, NOD2, REG1. cGAS is nothing but cytosolic GMP, AMP synthetase, which will activate cGMP. This in turn will go on to activate STING. These names you will hear 2 or 3 times more as we will be discussing interferonopathies in another 15 minutes from now. So my presentation will be slightly long. I'm going to interfere with your fun break. So you'll have to bear with me. So this is how, the DNAs are getting recognized, the RNAs are getting recognized. And why we need to know these pathways is at each of these steps, if there is any defect, it can lead to consequences. Either more infections, more severe infections, or it can result in autoimmunity. Similarly, the lipopolysaccharides activates the TLRs, and then there are these downstream pathways which will ultimately result in recruitment of or activation of first and the second waves, that is interferon IRF3, which is the first wave, and IRF7, which is the second wave, and will result in formation of interferon alpha and interferon beta. Now what happens next? Once these have got activated, the interferons are formed. They are going to act on these neighboring cells. They are going to act on their own cells. And how do they do? These are the type 1 interferon receptors. They are loosely, attached at the cell surface. Once an interferon comes, they will dimerize, they will come close, they will get adhered. And the JAKs which were, lying lazily, they suddenly get activated and they will get phosphorylated. Once they get phosphorylated, docking sites will be created for STATs, JAK-STAT. I'm sure a lot of us are hearing now about JAK inhibitors, and the role of these JAK inhibitors we will learn as we will understand more about interferons. So once a docking site is created, the STAT gets activated. Once that has got activated, it has some homework. It will go inside the nucleus and it will start stimulating genes which are responsive to interferons. And they have multiple of these complexes. So depending on which STAT is getting activated, which pathway is getting activated, a response by that particular cell is being determined. If it is this classical STAT1, STAT2, it results in some response elements which are getting activated. PI3 kinases has multiple effects. So once they have got activated, depending on the cell type, depending on what downstream pathway is getting activated, there are a lot of multitude of effects that we see. It's very difficult at times to remember which JAK is associated with which cytokines. I used to remember it that let's assume all have JAK1 and JAK3 with some important exceptions. IL-12 family has JAK2 and TYK2. IFN gamma and IL-6 are like, we are the supremes. A lot of people talk about us. So they take JAK1 and JAK2. And interferon alpha and beta, they need JAK1 and TYK2. Why we need to know? Because if we know the disease pathogenesis, if we know which of the interferon or the downstream pathways more implicated there, we can use therapy in that fashion. So now having learned all this, we should also know how we are killing the virus. इतना पढ़ा है तो virus कैसे मारना है वो भी सीख लेते हैं. So we understood how they were getting recognized. We understood how this genes inside were getting stimulated. And once that has happened, they are going to activate multiple downstream pathways in that particular cell, which ultimately leads to inhibition of viral protein synthesis, degradation of viral RNA, and inhibition of viral gene expression and virion assembly. And these are the pathways. I think we did not really remember all of these, but these are the terms, like myxovirus resistant GTpases, they, inhibit viral gene expression and virion assembly. So this is what is happening inside the cell. And we always have to prove, in science, this burden is always there. So people have done experiments where they have done knockouts of interferon alpha, gamma, and shown how the impact of these knockouts have affected the mice. So for example, this wild type mice where the interferon expression was happening in a desired way, when they were infected with VSV virus, there was no issue. But in this knockout model, where the interferon alpha was knocked out, you see a lot of staining of there are a lot of replication of the VSV virus. So to summarize, this is what we have talked in last, I think, 10 minutes. The cells, once they get infected, the patterns are getting recognized, the interferon alpha and beta are being formed. Inside the nucleus, they will stimulate various other genes, resulting in activation of these three pathways, which will ultimately result in killing of the virus. But is that enough? Is that the end? Do we need any other cells to come into play? ये वायरस के लिए बहुत है इतना. Does the adaptive arm get activated, or do other cells come into play? Yes, they do, right? So the initial response is by type 1 interferon, but then gradually the NK cells and the adaptive arm are getting activated. And how are they getting activated? What does interferon alpha does is, those are the T cells which are there in the lymph nodes, they express this receptor called as S1PR1, which is sphingosine 1, which is basically helps them to move out of lymph nodes. Interferon alpha does not allow them to move out of lymph nodes. They decrease the expression. And at the same time, they make sure that the DC cells are coming to lymphoid cells. As a result of which, there is a crosstalk between the innate arm and the adaptive arm. The T cells are inside. They are getting, converted into T follicular helper cells. They will interact with B cells. They will form antibodies. And that's how we have so-called those SARS-CoV-2 antibodies being formed, which acts against the viruses. And at the same time, they are also activating the NK cells and increasing the MSC1 expression, because you want the antigens to present these, process these antigens and present it, and you want them to be killed. Now, virus bhi ladega. It's not like, everything is good. So virus has also its own mechanisms by which it is trying to fight it out. And that is why we sometimes get mild infection and severe infection depending on that battle. For example, the influenza virus, they have their own armamentarium like this P58 IP mediated. They are going to inhibit the PKR that we saw in that downstream pathway where they're trying to inhibit. Similarly, hepatitis C has its own armamentarium in form of NS5A and E2 proteins. They interact with PKRs and inhibit these downstream pathways. So the question now, after having learnt all this, we need to answer is, how does someone get serious COVID? How does some infection can precipitate cytokine release syndromes or MAS? I think I'll skip this part. So we also need to ensure that interferon does not activate each and every cell around. ये भी बहुत ज़रूरी है, नहीं तो it will be lot of inflammation. So every cell type is destined in some way of getting activated. They have these mechanisms, like for example the expression of STAT. This is not going to happen in all the cells. So it's not that interferon alpha will go and start stimulating all the cells in the body, and that might result in a lot of issues. So there are negative regulators which help us to take care of this inflammation. We should not leave poor type 2 and type 3. This is simple. Type 2 interferon is nothing but interferon gamma. They again have their own receptors, JAK1, JAK2. They have their downstream pathways, and they are important for cell programming, for antigen presentation, cell recruitment, so on and so forth. And these are the cells which secrete interferon gamma, ILC1s, TH1, and cytotoxic. Similarly, we have IFN lambda, which is type 3. They are very important for immune defense at epithelial barrier surface. And recently, they have also found that these are the ones which help in T-cell education in the thymus. This is how the lambda looks like.
You have enough time. Don't rush. You have 10 more minutes.
Okay.
You have 10 more minutes. So
I have another 20 minutes of presentation left. So type 3 interferon, so I'll get a little slower. They have again their own receptors. Now we have understood this morphology, right? At the cell surface, there is downstream JAK, TYK2. They are going to activate STAT1 here. So that is what I was talking about. They have their own downstream pathways, and hence they have their own roles. So in fact, type 3 interferon has been found to be, more anti-inflammatory. If they are able to, what they do is they decrease the reactive oxygen species which are produced by neutrophils. They decrease the net formation which Sanket was talking about. So this is what interferon lambda or the type 3 lambda does. Very slow but a longer sustained response. Then why do we get chronic infections? If we have such good mechanisms, why is it that some of the patients have chronic viremia? The infections tend to go a little longer as compared to, you must have come across people who had COVID for 1 month, antibodies were coming positive, they had some mild symptoms. The reason is there are escape mechanisms even there. Say for example, if we saw there were multiple STATs which were getting activated, if it is STAT1 which is getting activated, there are less T and NK cells. As a result of which, we are not able to hold the virus for long. If you are not able to hold the virus, there is increased hyperresponsiveness, which is step number 2. So that results in chronic viremia. And at the other end, we have autoimmune disease. What are these cells doing? The influenza virus is making these cells to produce type 1 interferon. And these type 1 interferon are making these monocytes to express TNF-related apoptosis-inducing ligand, which are going to kill these cells around, the epithelial cells, as a result of which lots of self-antigens will start getting exposed. Now this happens because there was some problem at the check, there was some problem at the clearance, or there were different STATs which were getting activated. So these interferon alpha and beta can mediate upregulation of these apoptosis-inducing proteins, as a result of which there will be tissue damage. And with tissue damage, it can either lead into chronic viremia, the infection may prolong longer, or it may set off an autoimmune disease also. There are certain non-viral functions also. This is just for the completion's sake. All these interferons, they have some role, some protective role against these bacteria which are in green. Out here, they may be detrimental because they result in more damage. So these are, various mechanisms by which these interferons have some non-viral roles also. This is just for your knowledge, because interferon is just not purely about antiviral immunity. Now we will see the applied aspects. This is the last 1/3 probably. If there are no interferons, there is increased infection. So we will learn about MAS, we will learn about severe COVID. If there is too much interferons, we have interferonopathies. And if it is dysregulated, we have autoimmune diseases. So look at these circles. Interferon gamma receptor 1 and 2, we saw the one of the figures. If these pathways are getting affected, there is a disease called as MSMD, where there is Mendelian susceptibility to Mycobacterium. Because the interferon gamma is not getting produced as it should be, as a result of which these patients are more predisposed to infection with TB. Similarly, if you look on the right side of the figure, this is what we had gone through, right? TLR3, I think it sounds familiar now. TLR3, TLR7, 8, 9, downstream we had TRIF, TRAM, then we have tang binding kinases. So if any of these pathways are getting affected, the cell is not able to mount a good antiviral response. There are no interferons there, and they are more predisposed to infection. So for example, the blue colored ones are implicated in herpes simplex, encephalitis. And this is the list, you know, interferon alpha receptor 1, if it is defective, it can result in these kind of infections. If STAT is defective, more influenza, more severe influenza. That's what we keep hearing. Some patients are getting more serious infections. Life-threatening COVID-19 has been identified in patients who had mutations in TLR3, interferon lambda, TICAM1, which was nothing but same as TRIF, and so on and so forth, you know. So what happens if there is too much of interferons? We saw this figure, you would be able to recollect. What if this is getting activated a lot? What if this is getting activated? What if STING is super active? We get something called as interferonopathies, right? And this is, let's take this figure one by one. This is Aicardi-Gouttières syndrome, which is characterized by fever, chilblains, CNS inflammation, and basal ganglia calcification. The TREX, which has some work to degrade the nuclear material, is defective in these patients. If that is defective, what happens is lots of nuclear material is lying inside the cell. They are going to activate C-gas, which is nothing but cGMP, AMP synthetase. This in turn will activate the downstream pathway, and there will be induction of interferons. Something similar happens with SAVI. The STING gets super active. It is working on its own, you know. It's continuously producing interferons, and that's where we get this disease called as SAVI, which is characterized by rashes that can ulcerate and worsen in cold, ILD, and lots of autoantibodies. Something similar happens on the other side. You know, MAVs, they sense the cytosolic dsRNAs. If there is an activating mutation of this molecule, it results in increased production of interferons. And then there are some inhibitory molecules like USP18. If they are defective, again, that will result in more interferons being produced. So what is this cytokine storm that we talk about a lot? COVID-19, we talked about cytokine storm. Haemophilus influenza, we talk about cytokine storm. Cytokine release syndrome, something we see with CAR T cells, and macrophage activation syndrome. Now, if you look at MAS, it is this interferon gamma which plays the most important role. And that is why we had this beautiful, you know, these have not yet been published, but Empalizumab, which is anti-interferon gamma monoclonal antibody, was tried in these subset of patients. And if you look at each of these figures, there is an improvement in the platelet counts, there is a decrease in the D dimer, the platelets have improved, right? The amount of interferon gamma which were measured at the beginning have started to come down. That's all because of this molecule called as Empalizumab. Now, this is only possible because we need to understand the pathophysiology. And something similar has also been done in COVID. People have done genetic studies and found that those patients who had life-threatening COVID had more of mutations in interferon alpha receptor 1. They were not able to express it properly. Similarly, IRF7, TLR3. As a result of this, what happens is, I'll come to this figure first. What happens is, there is inadequate type 1 interferon response. Most of these viruses are able to reach the lung. And that's where they are now mounting a hyper-inflammatory response. We are not able to get rid of the virus at the first step. And this is what happens. So again, people have shown the highest odds ratio with these names will now sound your bells. TLR3, TICAM, TBK1. अभी नाम आएगा तो डर नहीं लगेगा because now we have come across these words, right? So a mutation in any of these downstream pathways is resulting in more severe life-threatening COVID. Something similar which also happens with other viruses. Now I'll just wind up with what happens with interferons in lupus. That's another interferon disease. So in a patient who is genetically predisposed, they are not able to clear these viruses. The damage is so much that lots of nuclear material is getting exposed, is coming out. As a result of which, there is interferon signature which can be seen in patients with lupus. And that is why people have tried, so I'll come to this figure. People are trying to target these pathways. The interferon producing cells are producing alpha, beta, and gamma. And there are anti-interferon alpha therapies which people have tried targeting these molecules. Cefalimumab, rontalizumab, but unfortunately, though they were able to show a decrease in the interferon signature in the tissues of these patients, it did not reach their primary endpoints. However, the anifrolumab, there are 2 trials which have already been published, looks slightly more promising. This is directed against the receptor. And people are now playing with the idea of using an interferon alpha chenoid vaccine, which when given to a patient, will result in production of anti-interferon alpha antibodies, which will take care of enterophenopathies, lupus, so on and so forth. And then we also learned about the downstream pathways, right? I think there is pin drop silence now. I'm just about to finish. So downstream pathways, we learned about how JAK1, JAK2, TYK2 were getting implicated. So all those diseases where you understand the pathogenesis, if you are aware that interferons are playing a role, you might yourself get an idea some of these molecules may work there. And there are lots of studies which are happening, as you can see, lots of phase 1, phase 2, phase 3 trial which are happening in lupus, RA, Sjogren, systemic sclerosis, dermatomyositis, because of the interferon signature which is seen in these diseases as well. And besides that, the interferons have been even tried in hepatitis B, because we have learned interferon helps us in fighting viruses. So people have tried using these interferons in the hepatitis B, hepatitis C, though now the treatment for hepatitis C has completely changed. People have tried interferon beta in multiple sclerosis because it has its anti-inflammatory effects. It helps in T cell skewing. It inhibits MMPs. It improves the blood-brain barrier, and so on and so forth. So the confused summary at the end is, this is how the family looks like. The type 1, type 2, type 3. These are the types of interferons. These are their role, and we learned how they were playing this role by looking at the downstream pathway. If there are no interferons, there are more infections. If there are too much interferon, you may get a cytokine storm. And if they are dysregulated, you get autoimmune diseases. Thank you.
Anyone wants to ask something? Anu, you want to ask something? Joe.
Amnesh, nice talk. As you said, in COVID, the outcome depended on. Can you just?
He's interfering. He's interfering.
He is interfering.
Yeah, he is my barrier. He's going to help me.
So the outcome or what happened? Yeah, what happened to the COVID? Outcome depended on the interferon response. Those who didn't have adequate interferon response had a more severe COVID. Right. That's now the key takeaway in COVID in these ones. Right. But none of these COVID trials didn't take well. They gave interferon. They didn't have a better outcome and all these things. So
yeah, that is something which is slightly, intriguing. Even in lupus, for example, I was talking about those interferon alpha antagonists. Probably the pathways, there are other mechanisms by which they are, this is all redundant, that's why that's a big problem. So probably it's not that easy, like I was telling. There are multiple pathways, there are other kind of cytokines which probably are getting activated. Targeting just one of these subtypes doesn't really help. Even in those genetic studies where they had done, it was only, though the odds ratio was really high, but the hypothesis was that they might be able to find some interferon-related defects in all of these patients, but they did not. Like for example, only in 3% in one of the study, only in 3% of these patients they found these defects. But yes, they were detrimental. But then they had another argument to it, that so far they were all in their 50s and 60s. Most of them were male. They were not infected by any other serious viral infection so far. So it is still very not clear whether they were, present to begin with, or it happened as the process was happening. So this is something which is very, very much not clear. That's why most of these therapies have not, but it at least gives us an understanding that this might be going wrong in these patients who have life-threatening COVID. What they actually found more was antibodies to interferon alpha in these patients. Almost 10 to 15% of these patients had antibodies which were working against interferon alpha, as a result of which they were getting more serious COVID. And that's why this thought came up, कि rituximab दे दे क्या इसमें, plasmapheresis, will that help? But then a better idea would have been giving them interferon beta, because these antibodies are predominantly against interferon alpha. So if you give them some other interferon, maybe that might work like in hepatitis B and C. But the practical utility is...
Thank you, Avi. So another question is, how do you think the interferon signature is going to change the way we practice? How is it important? How do we measure this interferon signature? Do you think it will come to clinics?
It sounds promising.
Can you just elaborate that for the crowd,
Yeah.
interferon signature, how
So,
is it measured?
so people, what they're trying to do is, most of these even antibodies or monoclonal antibodies which are coming up, they are realizing that all of these antibodies do not work in the same fashion as it should have been, or they are only able to take care of some subset of these patients. So what they needed to understand is to identify a group of patients where they will benefit more. So for example, what does the logical thinking say? Say for example, if you are using IL-17 inhibitors, तो जिन patients में IL-17 signature ज़्यादा होगा, maybe it will help them there. Similarly with interferon depleting therapies, if you want to use anti-interferon therapies, you might want to do assess interferon signature in these subset of patients, because not all lupus patients will be manifesting that kind of signature. So maybe these subsets are going to help you. So in near future, maybe in 10 years, you would start this. That is what precision medicine is going to be, right? Trying to identify the pattern of that disease in that particular patient. But having said that, it doesn't mean that all those patients who have that kind of signature will benefit in the same way because of the redundancies, because of the other pathways which get activated. So I think they may have a role, but at present I doubt, how much practical application will come out of it as of now.
Interferon gene
So,
signature.
no.
INSPIRE cohort for the less informed,
Yeah.
this is basically one of the largest cohort of lupus patients, around 4,000 lupus patients, which Avinash also is part of it.
Yeah.
So they have, are you doing that?
So we are not doing it. So this was not one of the objectives, but in one of the, so this has been proposed that we should look at, so we have around 2,503 patients as of now. We have stopped enrolling. This was a 5-year project. And we have SERA, DNA material of all these patients. Now the plan is, the more the ideas that the PIs get, the more we can work on. And one of the idea actually is to work on the interferon signature in these patients. कि सच में कितना है, and does it really correlate with the disease severity? Though patients who have more severe disease, will they have more more signature like that? At present, we are not looking at it, but that is one of the plans.
Can you explain how much of a role the dysregulated interferon plays in the initiation of disease and then on the sustenance of disease?
Right.
So, I think there is a difference, isn't there? At the initiation of disease, the interferon seems to play a bigger role.
Right.
And thereafter, for sustenance of the disease, it is not so dependent on the interferons for ongoing activity of autoimmune disease.
Right. So that's true. I think that's a very good point. So just we would expect the innate arm to get activated, and like people were talking about, why the innate arm was not studied so far, and it is the end result that we see most of the time. So yes, it plays a role to begin with. In the maintenance, ultimately, the adaptive arm does take over, but we still don't know in those patients who keeps getting intermittent flares, what is happening there. Probably in these subset of patients, the innate arm, the interferons might still be getting activated again, and the signatures might come high. And that is what one of the plan is, to look at these patients who are doing well, suddenly they start misbehaving, not with the doctor, but their disease. So whether the interferon signature is again getting activated in these subset of patients. Because I don't think it's a continuous thing. A part may get more activated, the other part may, lie dormant there. And once again the disease becomes active, the interferon signature may again.
My thoughts were that because of the limited response to anti-interferon antibodies during the course of the disease, that perhaps it has got a bigger role
Right.
in the initial phase of
So
the
you're
disease.
like trying to catch them early.
Disease early, just like
Yeah.
you catch RA early
Yeah.
and treat RA early. If you can catch lupus early,
Yeah.
perhaps we are not sifting the patients correctly
Right.
in order
Right.
to gauge their response.
No, I think if we are able to do that, the biggest problem with lupus is since how long these patients have been having this dysregulated immune system is very difficult to figure out. We learn about how these antibodies can predate clinical features, so-called that preclinical, and whether we should treat them that time is something very debatable because not all of these patients will go on to manifest lupus. So ultimately, dawa tab bhi milna hai, tab bhi milna hai. So we have to take a call.
Last question, young man. Yeah. Please introduce yourself before you ask a question. It will be great.
Hello, sir. Thank you for the lecture. I am Dr. Rahul, Assistant Professor, SDM Medical College. Sir. So, one thing was regarding the nomenclature of interferon, sir. This was a new thing for me. What I thought was interference is a property of viruses, how they interact within the same host. Like, if the host is already infected by a virus, the host reacts in a different way to another viral infection. So that property of interference, probably it was a protective mechanism through which some people, at least kids, were protected from severe COVID. That was a theory put forward, sir. So will the infection from a virus protect from another viral infection through this interferon mechanism? Does this theory hold good in this interferonopathies also? Regarding the severity of interferonopathies. If the host is more predisposed to a particular interferonopathy, the manifestations may not be severe as other subclasses of other interferonopathies.
Yeah.
Will this hold good too, sir?
So again, a good question. So to answer that, in fact, I had one of the slides on that too. Now the problem is nothing is black and white, right? To try to explain severe COVID, how did they explain was that the initial response that these patients were mounting was not good. The amount of interferons that they were producing was not good. As a result of it, there was more viremia, which was resulting in more inflammation later on. But on the other hand, if you have very good response, you might want to think that we might be protected from subsequent similar infection or any other infection. But people have actually shown that following a viral infection, the severity of bacterial infection was found to be much higher. Just an example, influenza ke baad, if a bacterial infection was happening, in fact, if you look at your cohort of patients or patients who die with influenza, you will realize that they had some secondary bacterial infection. Why? Because of a strong interferon response, all the other cells, there was some cross-firing happening. The T and B cells were already in an active mode. And they realized that the mortality was higher. So I think it's very difficult to say that a prior infection is how it's going to behave with a subsequent similar or some other infection. I hope I was able to make some sense.
Yes, sir. Some
sense.
Thank you, sir.
This MDFI myositis, like it has pathogenic role in the interferon pathway, and the clinical picture same as SAVI, like more of rashes and more of ILD. Like, can it be included in interferonopathies?
So, as of now, how the interferonopathies are getting classified is if you are able to find that mutation. So, MDF5 has been implicated, but I am not sure if in all those subset of myositis patients, have the people been able to show some genetic defect in these patients? If yes, they will probably present much earlier and they may have symptoms similar to interferonopathy and you might be able to classify it among them. But what is more important is identifying...
Anti-MDF5, it's not
Yeah.
MDF5, it's anti-MDF5 antibody, right? Yes. So, it is
No,
not
probably what he's trying to ask is in these subset of patients, if they have genetic mutation, so probably they will present in a different way and in earlier as compared.
Regarding MDF5, our current classification of disease is phenotype based. We are diagnosing myositis, finding the antibody, calling it MDF5. But as science progresses, sir has said, in next 10 years with multi-omics, everything is going to change. Our understanding of disease will change. Then we may have to reclassify many diseases. So it may be like a antibody associated interferonopathy. We don't know, we can have some new group. But classically, what we understand now is there has to be a genetic defect leading to high interferon signature to be called an interferonopathy, monogenic defect.
Yeah.
So if I can just add to what Dr. Saket just said, for MDA5, anti-MDA5 antibodies are there. And there's actually one school of thought that believes that the anti-MDA5 antibody goes and stabilizes MDA5. So when MDA5 is more stable in the cell, it is more active and generates more interferon, and the interferon signature increases. So you might have a point there that it can be reclassified in the future as an interferonopathy, but an acquired interferonopathy. That's the critical difference there.